It's a detailed step-by-step description of the standard patch clamp protocol. Patch clamp is a laboratory technique in electrophysiology that allows investigation of the electrical excitability of neurons and the functional properties and densities of ion channels. It is widely used to evaluate the toxicity of potential drugs, especially cardiotoxicity. Since its first conception, patch clamp techniques have undergone modifications into several configurations, which can record an individual ion channel or obtain the electric current of the whole cell or tissue slices. Cell-attached configuration. Considered the original patch clamp, the cell-attached patch clamp measures membrane potential from the electric current that passes an ion channel. In this configuration, the patch pipette forms a highly resistant seal with a membrane, called gigaseal, enabling picoscale measurement of the membrane potential. The cell-attached patch configuration does not require the electrode to penetrate the membrane, leaving the cell intact during patch clamp. For this reason, this type resembles the native physiological state the most. Whole cell configuration. In this configuration, the tissue or cells are immersed in the bath solution together with the bath electrode. To obtain the membrane potential, puncture the cell membrane with the patch pipette. The disrupted membrane allowed the pipette buffer to directly contact the cytoplasm, resulting in membrane potential measurement, or an instant current injection that compensates for the voltage difference between the intracellular and extracellular environment. This type of patch clamp is most similar to intracellular recording and is used for the overall assessment of ion channels distributed in the whole cell or tissue slices. It also permits the manipulation and control of the cytosolic environment through the pipette buffer. However, the whole cell patch requires a highly skilled worker who can avoid the drainage of cytosol that could inadvertently remove essential cytosolic factors. Outside-out configuration. Modified from the whole cell patch clamp, outside-out excised configuration measures an individual ion channel by rupturing the cell membrane. Here, the patch pipette is pulled away after it makes contact with the membrane, causing the membrane phospholipids to aggregate around the patch pipette. This lets users test the effect of extracellular factors directly on the ion channel. This configuration disrupts cellular cytoskeletons and requires skilled workers to manipulate the cell without completely letting the cytosol leak into the bath solution. Inside-out configuration. Similar to the outside-out excised patch, the membrane is punctured by pulling the patch pipette away after its initial contact with the membrane. Unlike the outside-out type, the patch pipette is briefly removed from the bath solution, leaving the patch with the bath-facing cytosolic side. The inside-out excised patch configuration necessitates a bath solution that resembles the cytosol to prevent cytosolic washout, making this patch clamp type suitable for testing the influence of a specific extracellular factor on the ion channels. Like the outside-out excised patch, the inside-out one also destroys the cytoskeleton network and requires a skilled worker to handle the experiment. Perforated configuration. Another whole cell patch modification, a perforated patch clamp records the whole cell membrane potential. The setup in the perforated patch is like the whole cell patch clamp, except an antifungal such as amphotericin B and nystatin is added to the pipette solution. It acts as a membrane perforating agent that only allows small ions to pass through the punctured membrane, blocking the loss of large cytosolic components. Here is a standard protocol of blind patch clamp. The required reagents are as follows. Here are the equipments required. The procedure is as follows. There are lots of ways that patch clamp assay can go wrong. Creative BioArray hopes to aid in troubleshooting when unacceptable results do occur. I hope this helps. Let me know if you need any more assistance.